Good afternoon from Norwich. I'm Ezgi. I'm working as a postdoc in Quadram and Orham Institutes, and I'm interested in increasing the resolution of microbiome profiling using both dry and wet lab techniques. And today I'm going to present you our Amplicon sequencing data analysis pipeline, LotuS2. Amplicon sequencing is still a widely used technique, and it's based on targeting a marker gene and amplification and sequencing of it. It's still widely used because it's a cost-efficient and high-throughput technique. Many of the childhood diseases have been successfully tackled by Amplicon sequencing, although some still remain. That's why, actually, Amplicon data analysis should be affordable for every lab using desktop computers and without specialized training. LotuS means less OTU Scripts, and it's designed to be a streamlined and easy-to-use pipeline, and it's possible to run LotuS2 in single command. For example, here's an example command from a LotuS2 run, and to be able to run it in single command makes it like it actually makes it with less user intervention. However, to complete the same analysis, I had to use between 17 to 38 commands with other pipelines. And a Galaxy Web interface for LotuS2 is also available, where you can upload raw reads and start the analysis. 63 different flags are available in LotuS2, and semi-automatic defaults are available for these flags, but also fine-tuning of the parameters is possible for the expert user. A path for the raw reads and a map is required to start the analysis in LotuS2. And the raw reads can be coming from different platforms like MySeq, HiSeq, PacBio, or 454. And LotuS2 also provides filtering defaults for each platform. And the mapping file can be also automatically generated by LotuS2. Common taxonomic databases such as Silva and GreenGenes or custom databases can be used in LotuS2. And there are 7 different clustering options. The primary outputs are a phylogenetic tree, count table, and a taxonomic table, which are merged in a phylosoc object, and this facilitates downstream analysis in R. Also, extensive log files are outputted, where detailed description of each step and also detailed reports on each step is summarized, and this makes the analysis transparent and reproducible. So this was an introduction for LotuS2, and now I want to demonstrate to you its computational power, but more importantly, I would like to show its precision and reliability. So to benchmark the computational power of LotuS2, we analyzed human gut and soil 16S datasets using LotuS2 and other Amplicon analysis pipelines. Each bar here represents the time needed to complete each step in the analysis, and this is in minutes. In both datasets, LotuS2 completed the analysis much faster. It was especially faster in the analysis of the human gut dataset because this dataset was non-demultiplex, and demultiplexing is the step where LotuS2 is especially faster. So the aim of a pipeline should be to incorporate as many as reads possible in the final output matrix, because if the user is investing money into sequencing, of course would like to use as much as of the data. However, this should be carefully balanced by filtering of technical artifacts, which can otherwise inflate the biodiversity. So based on this, higher number of retrieved sequence counts versus less biodiversity should be the optimal point for data usage efficiency. And to benchmark this, we compared percentage of reads retrieved by each pipeline versus the biodiversity generated at the end of the analysis. And LotuS2 was in the optimal point in data usage efficiency. So this means not only LotuS2 was completing the analysis in shorter time, but it was also using the most of the data. Next, we benchmark the reproducibility of the data produced by each pipeline. And to do so, we compared community composition of technical replicates in a human gut dataset. And by technical replicate here, I mean samples that are sequenced twice. And we expect less distance between the technical replicates, lower distance, and less is better because it means higher reproducibility. And overall, LotuS2 resulted in lower distance between the technical replicates. And we have done the same analysis in a different biome in the soil dataset, and LotuS2 still had the lowest distance between technical replicates. And in a third dataset from the same biome, but from a different marker gene, LotuS2 still had the highest reproducibility. Last, we interrogated its precision in a mock community, and we calculated fraction of risks that are assigned to a true positive taxa at genus level, as well as number of true positive, false positive, and false negative taxa, and based on those, calculated precision, recall, and F-score. LotuS2, especially using research and UParse clustering options, resulted in the highest precision, highest fraction of risks that are assigned to a true positive taxa. And for precision, LotuS2 still had the highest score. And for recall, a previous version of LotuS2, LotuS1, had the highest recall score. However, its precision was really low. And F-score is a summary score based on precision and recall. And LotuS2 had the highest F-score, so was winning the race to have the highest precision. So it was the most precise pipeline to reconstruct the mock community. This brings me to the end of my presentation, and I hope I was able to convince you that LotuS2 is doing a good job. And if you're interested in, you can install LotuS2 from its GitHub page, or you can use its Galaxy interface and start analysis by uploading the raw reads. And the preprint of LotuS2 is available now in bioRxiv and is under revision right now. And finally, I would like to acknowledge the developers of LotuS2 and members of Hildebrand Group, and thanks very much for listening to me.